everyone, today I'll be presenting about basic immunity. First, I will introduce our team members. My name is SAKINAH and my other members are ARIFAH, ALYA and KAMILIA. I will explain about biochemistry. Biochemistry looks at the chemical processes inside living things. It helps us understand how molecules like proteins and sugars keep the body alive and functioning properly. To compare, immunology focuses on how the body defends itself from infections and diseases. It explains how the immune system detects threats and protects us naturally. Next, the importance of the immune system. The immune system has several important roles. One of them is preventing diseases or infections. It can quickly detect harmful microorganisms and stop them from multiplying, preventing the infection from spreading to other parts of the body. The immune system also helps keep the body healthy and balanced. It makes sure our organs can function normally without being disturbed by infections or inflammation. Another key function is that the immune system creates memory cells. When the same pathogen enters again, these memory cells trigger a faster, stronger, and more accurate immune response, often stopping the infection before symptoms even appear. The immune system also plays a big role in maintaining homeostasis. It constantly communicates with the other systems such as the nervous system, endocrine system, and the circulatory system to keep everything stable. Now let's move on to the types of immunity. The first type is innate immunity, also known as the non-specific immunity. This is our first line of defense. It responds very quickly to pathogens, but it doesn't target specific microbes and it has no memory. Innate immunity has several components. We have physical barriers like the skin, mucous membranes, and epithelial layers that prevent pathogens from entering. We also have chemical barriers such as stomach acid, enzymes in saliva and tears, and antimicrobial peptides. There are also important cells involved in innate immunity. Phagocytes like macrophages and neutrophils engulf and destroy microbes. Natural killer or NK cells destroy virus-infected and abnormal cells. Dendritic cells capture antigens and help activate the adaptive immune system. Innate immunity also includes proteins and molecules like the complement system, which helps destroy microbes and promotes inflammation, and cytokines, which regulate and coordinate immune response. Next, we have adaptive immunity, which is specific. This type develops more slowly, but it targets particular pathogens and has memory. This means the response becomes faster and stronger upon repeated exposure. The main components of adaptive immunity are lymphocytes. B cells produce antibodies that bind to specific antigens, and some of these B cells become memory cells. We have also T cells. Helper T cells or CD4 plus cells activate B cells and other immune cells, while cytotoxic T cells or CD8 plus cells kill infected or abnormal cells. Moving on, the humoral immune response is part of the adaptive immune system, unlike the cell-mediated response which relies on T cells. The humoral response depends on the B lymphocyte and the antibodies they produce. Its main role is to neutralize pathogens that are freely circulating in body fluid, such as blood, lymph, and secretions before they can enter our cells. Let's break down the process step by step. First, antigen recognition. B cells carry special receptors on their surface. These receptors recognize and bind to specific antigens such as bacterial proteins or viral particles. Next, activation of B cells. Once the antigen is found, the B cell presents into the helper T cells using MHC class II molecules. The T cells in turn release cytokines like interleukin-4 and interleukin-6, which activate the B cell further. Next, clonal expansion and differentiation. The activated B cell multiplies and form two groups, which are plasma cell, which antibody factorizes, and memory B cells, which remains in the body for long-term protection. Last but not least is the antibody production. Plasma cells release large amounts of antibodies, also called immunoglobulins, that circulate in the bloodstream and neutralize pathogens. Now let's start with immunoglobulin G, also known as IgG. IgG is the main antibody found in our blood circulation. It plays several important roles in immunity. First, it binds to pathogens, which helps the immune system recognize and destroy them. It also activates complement proteins and promotes phagocytosis, which is when immune cells engulf and remove pathogens. Structurally, IgG is a monomer, meaning it has a single Y-shaped unit. One of the most important facts about IgG is that it is the only antibody that can cross the placenta. This allows mothers to pass immunity to their babies before birth. Because IgG remains in the body long term, it is also known as the antibody of memory. Next, we have immunoglobulin A or IgA. IgA is the main antibody found in body secretions, such as in mucus, saliva, and breast milk. Its main job is to prevent pathogens from attaching to epithelial cells in the digestive and respiratory tracts. In the serum, IgA is usually a monomer, but in secretions it becomes a dimer thanks to a joining chain. This structure helps it survive harsh environments like the stomach. Because it protects our body surfaces, IgA is considered an important part of our innate immune system. Now, let's look at immunoglobulin M or IgM. IgM is the largest antibody and is also found in circulation. It is especially good at activating complement proteins and is very effective at clumping viruses and bacteria together, which helps clear infections. IgM is the first antibody produced during a primary immune response. Structurally, IgM is a pentamer, meaning it has 5 Y-shaped units joined together. Interestingly, IgM can be formed by a fetus, but it cannot cross the placenta. Next is immunoglobulin E or IgE. IgE plays a major role in allergic reactions. It attaches to antigen receptors on basophils in the blood and mast cells in the tissues. When allergens bind to IgE, these cells release substances that trigger an immediate allergic response. IgE also provides protection against certain parasitic infections. Its structure is a monomer. Now let's move on to immunoglobulin D or IgD. IgD is found mainly on the surface of B cells. Its main function is to help in the activation of B lymphocytes, which is a key step in the adaptive immune response. Structurally, IgD is a monomer. Finally, this slide summarizes all 5 classes of immunoglobulins using the acronym KIMT. Next, antibodies play several important roles in clinical practice, not just in immunity but also in medical diagnosis and patient management. First, antibodies are used in diagnostic testing. Many laboratory tests, such as ELISA and rapid test kits, rely on antibodies to detect the presence of specific pathogens. These help confirm infections like dengue, HIV, influenza, and many others. Second, antibodies are essential in blood typing. In the ABO and RH systems, naturally occurring antibodies help identify a patient's blood group. This ensures that the correct blood group type is selected for transfusions and prevents dangerous transfusion reactions. Antibodies play a major role in how vaccines work and how the body protects itself from infections. Clinically, they are essential for preventing disease, reducing severity, and controlling outbreaks. First, in vaccination, antibodies are produced in response to the vaccine's antigen. These antibodies help the body recognize the pathogen early, so when a person is exposed in the future, the immune system can respond much faster and more effectively. Antibodies also help prevent the spread of infections worldwide by reducing a person's ability to transmit the pathogen, especially through mucosal immunity. Vaccination lowers overall disease circulation in the community. This contributes to herd protection and fewer outbreaks. They also reduce the risk of aggravated symptoms. Even if infection occurs, the presence of antibodies makes the illness much milder. Clinically, this means fewer severe cases, fewer hospital admissions, and lower chances of long-term complications. Antibodies further lower the risk of fatality due to infections. Many severe diseases such as measles, tetanus, and pneumococcal infections are far less deadly in vaccinated individuals because antibodies limit the spread and damage caused by the pathogen. Next, in terms of disease protection, antibodies neutralize viruses and bacteria, block them from entering cells, and help the immune system clear them out quickly. This prevents the infection from becoming serious and reduces the chances of complications. In addition, antibodies prevent recurrent disease and provide long-lasting protection. Memory B cells quickly produce antibodies during re-exposure, helping to prevent repeated infections and reducing the overall disease burden in hospitals and clinics. Clinically, antibodies are also used to help treat autoimmune diseases. Monoclonal antibodies can target and suppress harmful immune responses, improving symptoms and preventing further tissue damage. Lastly, antibodies help control infections even after exposure. Next, I will explain about the process of antigen recognition and antibody production. First, extracellular antigens are ingested and digested into antigen fragments by macrophage or APC. The antigen fragment is displayed with MHC on the outer surface of APC by forming antigen-MHC complex. Next, APC migrates to lymphatic tissue to present the antigen to T cell. Helper T cell receptors will recognize and bind to specific antigen fragments. APC secretes interleukin-1 to activate helper T cell. At the same time, antigen binds the receptor in the surface of a B cell. The antigen enters B cell through endocytosis and broken down to antigen fragments. Antigen fragments are then presented as antigen-MHC complex on the surface of B cell. The active helper T cell recognizes the antigen-MHC complex on B cell and secrete interleukin-2 to activate B cells. Next, the activated B cell undergoes proliferation and differentiation into plasma cell and memory B cell. Next, plasma cells secrete specific antibody. Antibodies enter circulation and bind with antigen-forming antigen-antibody complex. Finally, it is the end of our presentation about basic immunity. Welcome to our Q&A time. Let us open our Kahoot and play some games.